Welcome to the today's lecture. Today we would be discussing about adjuvants and cytokines in the vaccine. The main learning objective of this lecture is you will be getting to have a detailed insight about the need and concept of adjuvants, that why we need adjuvant material, where it is being used, what are the different types and classes of adjuvants and their mechanism of action. At the end of at the end of this lecture, you will be getting to understand the importance of adjuvants while doing vaccine preparation and their mechanism of action, and what kind of adjuvants or what types of adjuvants that we are currently using in different vaccine preparations. So starting with the introductory part, that what are adjuvants actually? First of all, non-living vaccines, especially those that comprise of very small molecules, they sometimes require the inclusion of the agent so as to enhance their efficacy. That means the vaccines, such kind of vaccines, which are not live vaccines, but they are prepared from some another means. In that case, to increase their effectiveness, we include some material or small molecules so that their efficacy can be enhanced. So these molecules are known as adjuvants that are basically added to enhance the effectiveness of the vaccines. These adjuvants, they can be of microbial origin, they can be synthetic, they can be endogenous preparations having different activities, but in the current scenario, most common adjuvants that we are using, those are aluminium or calcium salts that we regularly use in the vaccines prepared for the humans. The concept is actually these adjuvants, they basically enable the antigens, or you can say the vaccine material, whether it is in whole cell or some glycoprotein. So those antigenic molecules are they are controlled by adjuvants to be released in a very slow fashion so as to preserve the antigen integrity, and they can target the antigen presenting cell and cytotoxic T lymphocytes. So that is the main concept, so as to control the antigenic release inside the body. That means, in nutshell, we can say adjuvants are compounds that enhance the specific immune response against co-inoculated antigens. So that how our body will be mounting an immune response against the slow release of antigen, that is controlled by adjuvant material. Now the role of adjuvant. The first point is because they are basically involved in releasing slow release of uh, uh, antigenic material. So they play a very crucial role in improvisation of the immunogenicity of the antigens or vaccine, especially recombinant proteins, their epitopes, that most of them they sometimes lack the features of original pathogens, or they are quite weak immunogens. In that case, to increase their immunogenicity so that a strong immune response can be mounted, we add adjuvant. Second is, because of the addition of adjuvant, we can reduce the dose of antigens or the number of immunizations that are needed for protective immunity. You must have heard that in the previous times we used to have 4 or 5 doses of a particular vaccine, for example, hepatitis. But in the current scenario, we are taking only 2 doses. There are one dose that is a primary dose, and after that, there is a booster dose. So these all of these things are basically being are possible due to the addition of these adjuvants, so that the dosage or the number of immunizations can be reduced, but the efficacy can be increased. Because it is very important in improving the efficacy of vaccines in the newborns, the elderly and immunocompromised persons are also very safe while they take vaccines that are containing some adjuvant material. However, it also play a major important role in antigen delivery system for the uptake of antigens by the mucosa. So in that case also, because of the slow release, it is very easy for the mucosal system to uptake the antigen and mount the immune response. However, there are certain challenges because right now we are talking about these adjuvants that they are very effective, they improve the immunogenicity, They control the release of antigen, and they are very effective in newborns, or whether they are elderly patient or they are immunocompromised patients, they are effective. But the point is there are certain challenges that these adjuvants face. Yes, the first is adjuvant industry. To improve the safety and less reactogenic subunit vaccine. That means adjuvants should be of such type that it shall be safe, it shall not be reactive, and if you are using some subunit vaccines or recombinant vaccine, in that case it shall have safer effect, it shall not impose any adverse effect. Second is lower reactogenicity adjuvants. They came with the reduced vaccine efficacy. Now, if you are using such kind of adjuvant that is having least reactions or least reactivity, in that case, the efficacy of that very vaccine is compromised actually. So that is the major part. Next is safety and tolerability. Now, safety and tolerability are the critical regulatory issues that we normally face whenever we use the adjuvants. Because in most of the cases, the increased adjuvant potency is associated with in, increased reactogenicity and toxicity. Now, there are certain non-adverse reaction factors also. We shall use such kind of adjuvants that shall be cheap to produce, and they shall not induce any immune response against themselves. That means adjuvant shall not be recognized as foreign material by our body. But instead of that, they promote an appropriate immune response, that is cellular or antibody mediated immune response, depending upon the requirement for protection. So that is the main concept. Then is adjuvant affordability. It should be affordable, or it shall be in such a range that each and every person can, afford it, afford the vaccine that is containing the adjuvant. Next is it should be stable with the long shelf life and biodegradable also. That means its shelf life shall be quite a long, so that the vaccine shall not ruined out, and it shall be biodegradable. That means that adjuvant shall not impose any kind of harmful effects or environmental effects. It shall be biodegradable. The classical example of Edwax. It is an adjuvant. Next is whether adjuvant can cause some adverse effects. Yes, if the adverse effects that has been imposed by these adjuvants, they can be categorized into two reactions. One is local, another is systemic. Now, local injection. What happens in the local reaction? Suppose you have got some injection, and that injection at the site of injection site, the pain, inflammation, swelling, necrosis, lymphadenopathy, granuloma, ulcers, and generation of sterile abscesses sometimes happen due to the presence of adjuvant. And in case of systemic reaction means in that case acquired immunodeficiency, immunosuppression, eosinophilia, allergy, anaphylaxis, nausea, fever, or some arthritis or organ-specific toxicity, immunotoxicity are certain side effects that are imposed by adjuvants. However, the major problem that comes with the adjuvant production is the unsolved problem is how we can achieve the potency while we can avoid the reactogenicity and toxicity. The concept is, the point is, the main problem with adjuvant production is we shall have such kind of antigen that it shall be quite potent. Quite potent means it shall be very effective in enhancing the effectiveness or potency of the vaccine, or it shall be able to mount strong immune response, but it shall have no reactivity or no toxicity. So this is a major problem that comes while we are producing some adjuvant. This make their acceptance into the community for prophylactic vaccination, particularly in the pediatric difficult. Even we most commonly we use alum. Alum is it is an FDA approved adjuvant that we normally add in the vaccine preparations while the companies are preparing vaccines. They normally use alum. So even this alum, which is FDA approved, still has certain adverse effects like local injection site pain, inflammation, swelling, lymphadenopathy. So all of these adverse effects are basically we need to we can't overlook these adverse effects also. So we need to produce such kind of adjuvants. That is a major challenge that is being faced in the adjuvant industry. The major limitation with LM is inability to induce CTL. CTL means cytotoxic T lymphocytes. That is why the cellular mediated immune response is not mounted against certain specific antigens. However, there are certain factors that affect the antigen adjuvant selection. That how we can select that in this vaccine we need to add this adjuvant. The first thing is antigen. What kind of antigen we are using? Because as I told you that LM, which is an FDA approved adjuvant, it is not good for inducing cytotoxic T lymphocytes. So if LM is not good for CTL response, then what kind of antigen we can use? So depending upon the antigen nature, we need to decide the adjuvant. Second is species to be vaccinated, that what kind of species that we are going to vaccinate, because some of the adjuvants are like this, they are found to be safer in a one particular species, but they are not safe for another species. So in that case the species to be vaccinated is another important factor. Then is route of administration. Route of administration means how we will be administering that vaccine. In that case, whether the route is intramuscular, dermal, subcutaneous, or mucosal. Fourth point, important point is side effects or adverse effect. That means we must first of all weigh all the risks and benefits at that time. That whether the adverse effects are more or whether the positive effects are more. Now, having understood about adjuvants, their challenges, their need in the industry, why they are being added, different factors. Now, the major point is classification of these adjuvants. Basically, these adjuvants, they are classified according to their source, their mechanism of action, and physiochemical properties and administration route. Administration route mainly correspond to mucosal or in dermal. So depending upon these, they are divided into three groups. One is active immunostimulant, second is carrier, third is vehicle adjuvants. So in case of active immunostimulants, these are basically substances that increase the immune response to the antigen. However, in the case of carriers, there are certain molecules that being immunogenic proteins, they provide T cell help also. Then third is vehicle adjuvants. They being basically they are oil emulsions or liposomes that are prepared in the laboratory. They basically act as a matrix for antigen as well as they also mount and stimulate the immune response also. Then second classification is based upon the route of administration, that whether it is mucosal or perinatal. So classification based upon the physiochemical properties, that what kind of gel we are using, that depends upon the group of gel, based upon the tensoactive agents, oil emulsions, particulate, fusion proteins, LM salts, or many other mineral adjuvants also. So these are the basic challenges that we have discussed so far: toxicity, stability, bioavailability, cost, production difficulty, epitope modification, pre-existing immunity to career immunity. Toxicity, that means it shall not be toxic. Stability, it shall be stable. That is why it shall have a long shelf life. It shall not degrade or reduce the efficacy of vaccine. Bioavailability, because it shall be having such a potential that it control the slow release of antigen, so that the bioavailability time of the particular vaccine shall be increased. However, we shall use such kind of adjuvant that the cost is not so high, because each and every person can afford it. Then is production difficulty. Production difficulty means we shall not use such kind of adjuvant for which the production is first of all very tedious and time consuming, and after that the certain effects are quite difficult to control. Then fourth is epitope modification potential during formulation. That adjuvant shall not be of such type that while we are preparing vaccine formulation and it is able to affect on the vaccine material, that it is able to modify the epitope or modify the antigen. So that is a major challenge. Then is pre-existing immunity to carrier protein. Is there any report of pre-existing immunity towards the carrier protein that we are using? So there are certain adjuvant regulatory requirements that, first of all, whatever material we are going to use as adjuvant, we need to conduct preclinical studies, that is for toxicology studies prior to the phase 1 trial. Then dose and frequency of adjuvants to be increased during clinical studies so as to assess the safety complications, whether it is safe or not, whether there is any problem or not. So there are few adjuvant types that we are going to discuss. First is oil in water emulsion. Oil in water, that means adjuvant would be of such type that it would be having two phases. One would be oily phase, another would be water phase. That is why we are calling it as oil in water emulsion. Example is monatinite and adjuvant 65, lipovant. However, these all oil and water emulsion, they are quite irritating. However, they sometimes called local irritation, local inflammation. They sometimes induce chemotactic signals also. Because of that, all the macrophages are attracted to the site of injection. Sometimes invasion is happening, due to which rapid ingestion is happening. However, we use such kind of adjuvant types in cancer and HIV treatment. In that case, what happens is formation of depot at the injection site is formed. However, it enables the very slow release of antigen because of the presence of two phases. It can also stimulate the antibody producing plasma cells so that the particular antibody targeted against a specific antigen can be produced. However, there are certain limitations. For example, excessive reactogenicity, frequent side effects of emulsions, including inflammatory reactions, granuloma formation, or ulcers at the site of injection. Next is saponins. We can use many classes of saponins as vaccine adjuvant also. At first point is, till now we have discussed about adjuvants. Now we are talking about saponins as vaccine adjuvant. So a vaccine adjuvant, it is a substance that is added to the vaccine to increase the body's immune response towards a particular vaccines. And saponins, what are saponins? They are natural glycosides of steroid or triterpene molecules, and these molecules, saponins, steroids, glycosides, they are reported for many different biological and pharmacological activities in the past. So notably, we can use such kind of molecules, saponins, that can activate the mammalian immune response because they have the inbuilt ability to activate the immune response or mount the immune response against various infectious diseases. That is why the significant interest of vaccine industry is towards saponin to be used as vaccine adjuvant. However, the most widely used saponin-based adjuvants are, there are few examples of it. One is Quil A, and there are few derivatives of it, like QS21. It has been isolated from the bark of a plant, Qualagia spanoria molina, which have been evaluated in the numerous clinical trials, because this plant is basically medicinal plant and it is found to to be a major producer of saponin molecule. However, they are unique. Why we are using these saponins? Because their mechanism of action is quite, different, quite strong in mounting the immune response. Because they have the unique capacity to stimulate both the T helper 1 immune response as well as the production of cytotoxic T lymphocytes. That means it can side by side activate T helper cell also, so that it can activate B cell and produce antibody. On the other hand, it can also activate cytotoxic T lymphocytes so that the cytotoxic response can be mounted against the exogenous antigen. That is why they are ideal for use in subunit vaccines, and vaccines, those are directed against intracellular pathogens as well as for therapeutic cancer vaccines, because in most of the cases, in case of intracellular pathogens and for the cancer treatment, these vaccine adjuvants are being used for the vaccine preparation. However, there are certain limitations also. because sometimes whenever you use these saponins containing vaccines, you observe severe injection site pain is a major limiting factor whenever you use QS21 derivative of saponin as adjuvant. Next is there are certain microbial derived adjuvants also. That means bacteria derived adjuvants also. For example, lipopolysaccharide capsule, that is LPS, TDM, that is trihaloose dimycolate. However, both of these different molecules are being produced by bacteria and microbes, out of which there is one example of MPL, which is a chemically detoxified derivative of native lipid A, which is produced by Salmonella Minnesota R595 strain. However, this MPL, it is being used in the complex adjuvant formulations along with alum, which is an FDA approved, QS21, which is a saponin, liposomes, and emulsions. The main mechanism is, as I told you in the case of saponin, the main mechanism of action is that it trigger both the T helper cell as well as cytotoxic T lymphocytes. However, the bacteria derived adjuvants, they interact with the toll-like receptor 4 present onto the surface of macrophages, result due to which the release of inflammatory cytokines is triggered. That means once this molecule, this adjuvant, it interact with the toll-like receptor TLR present onto the macrophages, it will trigger the production of inflammatory cytokines, including tumor necrosis factor, interleukin 2, interferon gamma, because of which the T helper 1 response is mounted and the strong immune responses observed in case against the particular antigen. However, there are certain limitations again. There are consistency of preparation is very important, formulation and cost. That is a major limiting factor. Otherwise, it is very successful. Now, MF59, this is a replacement for CFA. However, this is found to be toxic for humans. However, the current version is now being used in influenza. And but it has been found that this MF59 as adjuvant, it is superior to alum in inducing antibody response to hepatitis B in humans and baboons. Another example is ISCOM, that is immunostimulating complexes containing saponins, a sterol or optionally a phospholipid. Basically, what it does, it mount a strong cytotoxic T lymphocyte response against the antigens like HIV, which is an enveloped virus. But the major limitation is cost, manufacturing difficulty, malaise, reactivity problem, and safety issues. These are the few complications that we normally observe against different range of adjuvants. However, there is another class, that is liposomes. Liposomes, what are liposomes actually? They are synthetic phospholipid spheres in which antigen is encapsulated. You can say your antigenic molecule is encapsulated inside a synthetic phospholipid layer and it acts as antigen delivery vehicle as well as antigen adjuvant also. Because the main mechanism is what happens is these liposomes, they fuses with the macrophages. What it does after fusion with the macrophages, due to the phospholipid sphere, they deliver the antigen to the cytoplasm in a very slow fashion. However, this is a very effective drug delivery and vaccine delivery method. There are certain limitations, again the cost and manufacturing difficulty, because first of all you need to prepare, you need to have pure antigenic preparation, then you need to construct the liposomes, then you need to use some drug delivery method or antigen delivery vehicle. So the cost and manufacturing difficulty is there. However, there is a minute problem of stability also. There are certain carbohydrates also that we can use as adjuvant. The example is Edwax. Basically, carbohydrate adjuvant. That means we will be using some carbohydrate moiety. So this Edwax, it is basically a nanocrystal of inulin. Inulin, it is a carbohydrate. Basically, it is a plant-derived polysaccharide, and this polysaccharide, it is made up of linear chain of fructose that is capped with the glucose. That means it is containing fructose and glucose molecules. And the main mechanism of action whenever we are using Edwax as an adjuvant, it enhances both cellular mediated immune response and humoral response without any reactive problem. That means it would be able to activate B cells so that plasma B cell can be formed and antibody can be produced. However, it can also activate CMI, that means it will activate cytotoxic T lymphocytes so that the cellular mediated immune response can be mounted with no or least reactive problems. The shelf life is quite long. There are no safety issues. They are not metabolized in mans, but they are secreted unchanged in the urine as fructose and small quantity of glucose. So these are the 14 factors whenever we use carbohydrate as adjuvants. There are certain salts that we have discussed, alum. Those are basically aluminium salts. They are inorganic salts that binds to proteins and they cause them to precipitate out. And once they bind to a protein and bring about their precipitation, what we can do whenever we are using these preparations in the vaccines, in that case we take a soluble antigen or antigenic preparation and we trap that antigen inside the LM gel. So once that trapped soluble antigen inside the LM gel, they release or they increase the duration of antigen interaction with the immune system because they can control the release of antigen inside the body. Because of the slow release of the antigen, the strong immune responses mounted. However, LM is the only adjuvant that is approved for use in humans because it induces both antibody response and cellular response. That means it activates both T helper 1 cells and T helper 2 cells, thereby mounting humoral response also and cellular mediated immune response also. That means it will be able to produce antibody against that specific antigen also, as well as it will be able to activate cytotoxic T lymphocyte also, so that the cytolytic activity can be imposed. However, there are certain limitations also because each and everything have certain limitations. So the limitation is severe local and systemic side effects like local injection site pain, sometimes inflammation is happening, swelling is happening, pus formation is happening, sterile abscesses is happening, eosinophilia is happening, myofasciitis is the problem. So there are certain different limitations of each and every adjuvant that we use in the vaccine preparation. There are certain polymeric microspherical adjuvants also. These are basically compact and biodegradable microspheres of lipids, and they are able to incorporate various varieties of antigens. That means they are broad spectrum. What we do, we prepare the microspheres of lipids because they are compact in size as well as they are biodegradable. That is why they are more commonly used. They control the timely release of the antigen inside the body. However, there are certain cytokines. Cytokines, these are glycoproteins, these are protein molecules that are produced by a vast range of cells. There are certain cytokines that can also act as adjuvants. For example, interferon gamma. They enhance the cellular mediated immune response through a variety of mechanisms. Another is growth factor, GM-CSF. This can be also used as an adjuvant. How we can go for the preparation of adjuvant formulation. However, the new adjuvant formulations that have resulted, they are basically designed from the mixture of different adjuvants in the same formulation. That means we are combining different classes of adjuvants so as to prepare one formulation. As a general rule, whenever we use two or more adjuvants with different mechanism of actions, they are basically combined. The main objective is to combine them to enhance the potency and the type of the immune response against the vaccine antigen. For example, you can take the example of LM salts. They are formulated in the vaccine industry. They are formulated in combination with other adjuvants such as lipid A to increase the immunogenicity. That means to mount a strong immune response, to increase the efficacy of the vaccine. So these are certain adjuvant formulations that we normally prepare before going into the vaccine preparation, so as to, because the basic concept is The vaccine should be such that it shall be effective, it shall have a longer shelf life, it shall not ruin out so easily, and it shall have a strong response, or it shall mount a strong response. These are the few MCQs pertaining to this lecture, that how the antibody product, how we can, which thing is basically used to stimulate the antibody production, what are the FDA approved adjuvants, why we need to use it, what are the challenges, what are the different types of adjuvants that we use in the vaccines, whether there are any saponin based vaccines, carbohydrate vaccines, carbohydrate based adjuvants, or what are their side effects, what are their limitations. So all of these things are currently discussed in this lecture. However, the concept is clear. In case these MCQs are also uploaded onto the LMS, in case there are any queries that can be addressed later in the chat box, or in the or by consultation after. Thank you so much for listening.